Ganong's Review of Medical Physiology, 25th edition. Kim E. Barrett, Susan E. Berman, Scott Boyton, Haddon L. Brooks. Ganong's Review Medical Physiology, 25th edition. Kim E. Barrett, PhD, Distinguished Professor, Department of Medicine, Dean of Graduate Division, University of California, San Diego, La Jolla, California. Susan N. Berman, PhD, Professor, Department of Physiology, Toxicology, State Michigan University, East Lansing, Michigan. Scott Boyton, PhD, Professor, Physiology and Cellular and Molecular Medicine, Arizona Respiratory Center. Bio5 Collaborative Research Institute, University of Arizona, Tucson, Arizona. Hadwin L. Brooks, PhD, Professor Physiology and Pharmacology, Professor of Medicine, University of Arizona, Tucson, Arizona. M.C. Graw-Hill Education, New York, Chicago, San Francisco, Athens, London, Madrid, Mexico City, Malin, New Delhi, Singapore, Sydney, Toronto. Dedicated to William Francis Ganong. Key features of the 25th edition of Ganong's Review of Medical Physiology: a concise, up-to-date, and clinically relevant review of human physiology, provides succinct coverage of every important topic without sacrificing comprehensive or readability. Reflects the large research and developments in the areas of chronic pain, reproductive physiology, and acid-based hemostasis. Incorporate examples of clinical medicine to illustrate important physiologic concepts. Section introduction helps you build a solid foundation on the given topic. Includes both end-to chapter and board-style review questions. Chapter summaries ensure retention of key concepts. More clinical senses and flowcharts than ever, along with modern approaches to therapy. Expanded legends of each illustration so that you don't have to refer back to the text. Introductory materials cover key principles of endocrine regulations in physiology. More than 600 full color illustrations. End of chapter review question helps you access your comprehension. Clinical case add real world relevance to the text. About the authors: Cam E. Barrett. Cam E. Barrett received her PhD in biological chemistry from University College London in 1982. Following postdoctoral training at the National Institute of Health, she joined the faculty of the University of California, San Diego School of Medicine in 1985, rising to the rank of Professor of Medicine in 1996, and was named Distinguished Professor of Medicine in 2015. Since 2006, she has also served the University of Dean of Graduate Division. Her research interests focus on physiology and pathology of the institute epithelium and how it functions. It altered by commensal probiotic and pathogenic bacteria, as well as in specific disease states such as inflammatory bowel disease. She has published more than 200 articles, chapters, and reviews, and has received several honors for her research accomplishments, including the Bowditch and Davenport lectureships from the American Physiological Society of the degree of Doctors of American Physiological Society degree of Doctors of Medicine Science honors in cause from Queen's University, Belfast. Susan A. Berman received her PhD physiology from Loyola University School of Medicine in Maywood, Illinois. Afterwards, she went to Michigan State University (MSU), where she is currently a professor in the Department of Pharmacology, Toxicology, and other neuroscience program. Dr. Berman has had a career-long interest in neurocontrol of cardiorespiratory function, and with an emphasis of the characterization and origin of naturally occurring diseases of symptoms and pharynx nerves. She was a recipient of a prestigious National Institute of the Health Merit Method of Extant Research in Time Award. She is also a recipient of an Outstanding University Women Faculty Award from the MSU Faculty Professional Women's Association. Scott Boyton Scott Boyton received his PhD in genetics and cell biology from Washington State University of Pullman, Washington, where he received an interest in cellular signaling. He fostered his interest at University of California, Los Angeles, where he focused his research on second messengers and cellular physiology of the lung epithelium. Hadwell D. Brooks received her PhD from Imperial College, University of London, and a professor in the Department of Physiology and Pharmacology in the University of Arizona, UA. Contents Preface. Section 1, Cellular and Molecular Basis of Medical Physiology 1, General Principles and Energy Production in Medical Physiology. Overview of cellular physiology and medical physiology. Immunity, infection, inflammation, excitable tissue, nerve, excitable tissue, muscle, sympathetic and junctional transmission, neurotransmitters and neuromodulators. Section 2, Central and Peripheral Neurophysiology, somatosensory neurotransmission, touch, pain and temperature, vision, hearing and equilibrium, smell and taste, reflex and voluntary control of posture and movement, autonomic nerve system, electrical activity of the brain, sleep-wake state, and circadian rhythms, learning, memory, language and speech. Section 3, Endocrine and Reproductive Physiology, basic concept of endocrine regulation, hypothalamic regulation of hormonal functions, the pituitary gland, the thyroid gland, the adrenal medulla and adrenal cortex, hormonal control of calcium phosphate metabolism, and the physiology of bone, reproductive development and function of the female reproductive system, function of the male reproductive system, endocrine function of the pancreas, and regulation of carbohydrate metabolism. Section 4, Gastrointestinal Physiology, overview of gastrointestinal function and regulation, digestion, absorption, nutritional principles, gastrointestinal motility, transport and metabolic functions of the liver. Section 5, Cardiovascular Physiology, origin of heartbeat and electrical activity of the heart as a pump, blood as circulatory fluid, and the dynamics of blood and lymph flow, cardiovascular regulation mechanisms, circulation through special regions. Section 6, Respiratory and Physiology, introduction of pulmonary structure and mechanics, gas transport and pH, regulation of respiration. Section 8, renal physiology, renal function and maturation, regulation of extracellular fluid composition and volume, acidification and urine bicarbonate excretion. Preface from the authors. Once again, we are dedicated to launch a new edition of Ganong's Review of Medical Physiology 25th. The authors have attempted to maintain the highest standards for excellence, accuracy, and pedagogy developed by Fran Ganong over 46 years, during which he educated countless students worldwide with this textbook. Recognizing the pivotal and increasing role of graphical materials in effective medical education, our goal for this new edition was to undertake a thorough overhaul of our program while also making important and timely updates to the text. The vast majority of the figures in this edition has been revised or are wholly new to aid in understanding across contact areas. We have used consistent coloring and diagrammatic schemes which possible to depict comparable structures, cell and organs. We have also included an increased number of cartoons and conceptual diagrams as well as flowcharts to promote learning of the integrated material of the defines physiology. Overall, we hope that the updates to the volume engage the student and make understanding and assimilation of the material a more pleasurable task. We remain grateful to the many colleagues and students contract who contacted us with suggestions for clarification and a new material upon receiving the 24th edition. This input helps us to ensure the text is as useful as possible, although the responsibility of any errors, which are almost inevitable, is a project of the scope remains with the author team. Nevertheless, we hope that you enjoy the fruits of our labors and the new material in the 25th edition. The edition is a revision of the original work of Dr. Francis Ganong. Section 1, regular molecular basis of medical physiology. Units for measuring concentration of solutes. In considering the effect of various physiological important substances and the interaction between them, the number of molecules, electrical charges, and particles of a substance per unit volume of a particular blood body fluid are often more meaningful than simply the weight of the substance per unit volume. For this reason, physiologic concentrations are frequently expressed in moles, equivalents, or osmoles. Moles. A mole is the gram molecular weight of substance. That is the molecular weight of the substance in grams. Each mole consists of 6 to the power 23 moles. The molecule mole is 1/1000th of a mole, the micromole. Thus, NaCl 23 grams plus 35.5 grams equal to 58.5 grams is 1 mole. The molecular weight of a substance is the ratio of the mass of a molecule of the substance to the mass 1/12th of the mass of an atom of carbon 12. Because molecular weight is a ratio, it's dimensionless. The delta D is a unit of mass equal to 1/12 the mass of an atom of carbon-12. The kilodalton is a useful unit for expressing the molecular mass of proteins. Thus, for example, one can speak of a 64 kilodalton protein or state that the molecule mass of the protein is 64,000 dalton. However, because molecular weight is a dimensionless ratio, it is incorrect to say that the molecular weight of a protein is 64 kilodalton. Section 1, Cellular and Molecular Basis for Medical Physiology. The detailed study of physiology system structure and function has its foundations in physical and chemical laws and the molecules and cellular makeup of each tissue and organ system. This first section provides an overview of the basic building blocks of that provide the important framework for human physiology. It is important to note that these initial sections are not meant to provide an exhaustive understanding of biophysics, biochemistry, or cellular and molecular physiology. Rather, they are to serve as a reminder of how the basic principles of these disciplines these disciplines contributes to medical physiology discussed in later sections. In the first part of the section, the following basic building blocks are introduced and discussed: electrolytes, carbohydrates, lipids, fatty acids, amino acids, protein, and nucleic acids. Students are reminded of some of the basic principles of building blocks of biophysics and biochemistry and how they fit into the physiologic environment. Examples of direct clinical applications are provided in the clinical boxes to help bridge the gap between building blocks, basic principles, and human physiology. These basic principles are followed up with a discussion of the generic cell and its components. It is important to realize that cell is the basic unit within the body, and it is the collection and the fine-tuned instructions among the between these fundamental units that allow for proper tissue, organ, and organism function.
Yeah. Second part of the introductory system. Introductory sections. We take a cellular approach to lay a groundwork of understanding groups of cells that interact with many of the system discussed in future chapters. The first group of cells presented contributed to inflammatory reaction in the body. These individual players, their coordinate behavior, and the net effects of the open system of inflammation in the body are discussed in detail. The second group of cells discussed are responsible for the excitatory responses in human physiology and include both neuronal and muscle cells. A fundamental understanding of the inner workings of the tissue cells and how they are controlled by their neighboring cells helps the student to understand their eventual integration into the individual system discussed in later sections. In the end, this first section serves as an introduction, refreshers, and quick sources of material to best understand system physiology presented in the later sections. For detailed understanding of any of the chapters within these sections, several excellent and current textbooks that provide more in-depth reviews of principles of biochemistry, biophysics, and physiology, biophysics and cell physiology, muscle and neural physiology, and provide as resources at the end of each individual chapters. Students who are intrigued by the overview provided in this first section are encouraged to visit this text for a more thorough understanding of these basic principles.
General principle and energy production in medical physiology. Chapter 1, objectives. After studying this topic, you should be able to define units used in measuring physiological properties, define pH and buffering, understand electrolytes, and define diffusion, osmosis, and tonicity. Tonicity. Understanding. Define and explain the significance of resisting membrane potential. Understand in general terms the basic building blocks of cell, nucleotides, amino acids, carbohydrates, and fatty acids. Understand higher order structure of the basic building blocks DNA, RNA, protein, and lipids. Understand the basic contributions of the basic building blocks to cell structure, function, and energy balance. Introduction.
In unicellular organism, all vital processes occur in a single cell. As the evolution of molecular organs progressed, various cell groups organized into tissue and organs have taken over the particular functions. In humans and other vertebrate animals, these specialized cell groups include a gastrointestinal system to digest and absorb food, a respiratory system to take up oxygen and eliminate carbon dioxide, a urinary system to remove waste, a cardiovascular system to distribute nutrients, oxygen, and the products of metabolism, a reproductive system to perpetuate these species, and nervous and endocrine system to coordinate and integrate the functions of the other system. This book is concerned with the way these systems function and the way each contributes to the function of the body as a whole. This first chapter focuses on a review of the basic biophysical and biochemical principles and the introduction of the molecular building blocks that contribute to cellular physiology. General principle: the body of organized solutions. The cell that make up the bodies of all but the simplest molecular animals, both aquatic and aquatic or terrestrial, exist in an interstellar sea of extracellular fluid and enclosed within the integument of the animal. From this fluid, the cells take up oxygen and nutrients into it. They discharge metabolic waste products. The extracellular fluid is more dilute than present day seawater, but its composition closely resembles that of the primordial ocean in which presumably all life originated. In animals with a closed vascular system, the extracellular fluid is divided into the intercellular fluid, the circulating blood, plasma, and the lymph fluid that bridges these two domains. The plasma and the cellular elements of the blood, principally red blood cells filled with vascular system, together they constitute the total blood volume. The intercellular fluid is the part of the extracellular fluid that is outside the vascular and lymph system, bathing the cell. About one third of the total body weight is extracellular, the remaining two thirds of the intercellular fluid. Inappropriate compartmentalization of the body fluid can result in edema. In the average young adult male, 18% of the body weight is protein related substance and 7% mineral, 15% is fat.
Clinical Box 101. Edema. Edema is buildup of body fluids within tissues. The increased fluid is related to an increased leak of the blood or and the lack of blood or and reduced removal of the lymph system. Edema is often observed in the feet, ankles, and legs, and but can happen in many other areas in the body. Response to disease including those of the heart, liver, lung, kidney, and thyroid. Thyroid highlights the best treatment for edema includes reversing the underlying disorder, thus proper diagnosis of the cause of edema is primary first step in therapy. More general treatments include restriction, dietary sodium to minimize fluid retention, and using appropriate plate diuretic therapy. The remaining 60% is water. The distribution of this water is shown on figure 11a. The intercellular compounds of the body water accounts for about 40% of the body weight and the extracellular components for about 20%. Approximately 25% of the extracellular components is the vascular system, plasma 5% of the body weight. Plasma 5% of the body weight. And 75% outside the blood vessels. Interstitial fluid 15% of the body weight. The total blood volume is about 8% of the body weight. Flow between these compartments is tightly regulated. Units of measurements: concentration of solutes. Water, electrolytes, and acid base. The water molecule is an ideal solvent for physiologic reaction. H2 has a dipole moment where oxygen slightly pulls away electrons from the hydrogen atom and creates a charge separated that makes the molecule polar. This allows water to dissolve a variety of charged atoms and molecules. It also allows the H2O molecule to interact with other H2O molecules hydrogen bonding via hydrogen bonding. The resulting hydrogen bond network in water allows for several key properties relevant to physiology. Water has a high surface tension. Water has a high heat of vaporization and heat capacity. And 3. Water has a high dielectric constant. In layman's term, H2O is an excellent biologic fluid that serves as a solute. It provides optimal heat transfer and conduction of current. Electrolytes. A molecule that dissolvates in water to their cation and cation and anion equivalents. Because of the net charge of water molecules, these electrolytes tend to resonate in water. There are many important electrolytes in physiology, notably Na plus K plus Ca2 plus Mg2 plus Cl minus and HCO3 minus. It is important to note that the electrolytes and other charged compounds are unevenly distributed in the body fluids. These separations play an important role in physiology. pH and buffering. The remanence of a stable hydrogen ion concentration in body fluids is essential to life. The pH of a solution is defined as the logarithm of base 10 of the reciprocal of the H plus, that is. Figure 1. Organization of body fluids and electrolytes into compartments. Body fluids can be divided into intracellular and extracellular fluid compartments, ICF and ECF respectively. Their contribution to percentage of body weight based on a healthy young adult male slightly varies with age and gender. Emphasize the dominance of fluid makeup in the body. Transcellular fluid, which constitute a very small percentage of the total body fluids, are not shown. Arrows represent fluid movement between compartments. Electrolytes and proteins are equally distributed among the body fluids. This unevenly distribution is crucial to physiology. Pro- Protein, which tends to have a negative charge at physiologic pH. Now the clinical box 1 to 2 acid base Disorders. Therapeutic highlights: diffusion, osmosis, concentration of plasma, tonicity, Donan effect, plasma osmolality and disease, non-ionic diffusion, force action on ions, genesis of the membrane potential, energy production and energy transfer, biological oxidations, and nucleotides and nucleic acids, DNA. Therapeutic highlights: replication, mitosis, meiosis, RNA.
Dependent on correctly identifying the underlying causal process, especially true when this is especially true when mixed disorders are encountered. Treatment of respiratory acidosis should be initially targeted at restoring ventilation, whereas treatment for respiratory alkalosis is focused on the reversal of the root causes. Bicarbonate is typical use as a treatment for acute metabolic acidosis. An adequate amount of a chloride salt can restore acid-base balance to normal over a matter of days for patient with a chloride responsive metabolic alkalosis, whereas the chloride resistant metabolic alkalosis requires treatment of the underlying disease.
Charge the H plus in solution. Change the H plus in solution. In physiologic compounds, most acid or base are considered weak. That is, they contribute or remove rarely few H plus H hydrogen ions from solution. Body pH is stabilized by the buffering capacity of the body fluids. A buffer is a substance that has the ability to bind or release hydrogen ion solution, thus keeping the pH of the solution relatively constant despite the addition of considerable quantities of acid or base. Of course, there are a number of buffers at work in biologic fluids at any given time. All buffer pairs in a homogeneous solution are in equilibrium with the same hydrogen ion. This is known as isohydric principle. One outcome of this principle is that by assaying a single buffer system, we can understand a great deal about the biologic buffers in the system. When acids are placed into solution, there is dissociation of some of the components acid into its proton and free acid. This is frequently written as an equation Ha उभयमुखी H plus ion Ra minus. A unique feature of HCO3 minus is the linkage between its buffering ability and the ability of the lungs to remove CO2 from the body. Other important biologic buffers include phosphate and proteins. Diffusion. Diffusion is the process by which a gas or a substance in a solution expands because of the motion of its particles to fill all the available volume. The particles, molecule or atoms of a substance dissolves in a solvent are in continuous random movement. A given particle is equally likely to out of an area in which it is present in high concentration. The total number of particles removing to areas of lower concentration is greater. That is, there is a net flux of solute particles from an area of high concentration to the area of low concentration. The time required for equilibrium by diffusion is proportional to the square to the diffusion distance. The magnitude of the diffusion tendency from one region to another is directly proportional to the cross-section area across which diffusion is taking place and the concentration gradient or chemical gradient, which is the difference in concentration of the diffusion substance divided by thickness of the boundary. Franck's law of diffusion. Thus, J equals to minus dA delta C by delta X, where J is the net area of diffusion, D is the diffusion coefficient, A is an area, and delta C by delta X is the concentration gradient. The minus sign indicates the direction of the diffusion. What considering movement of molecules from a higher to a lower concentration is negative, so multiplying by dA gives a positive value. The permeability of the boundary across which diffusion occurs in the body vary, but diffusion is still a major force affecting the distribution of water and solutes. Osmosis. When a substance is dissolved in water, the concentration of water molecules in a solution is less than the pure water. Because of the addition of solutes in water results in a solution that occupies a greater volume than the greater water alone. If the solution is placed on one side of a membrane, there is a permeable permeable to water but not to solute. An equal volume of water is placed on the other. Water molecules diffuse down their concentration chemical gradient into the solution. This process, the diffusion of solvent molecules into the higher region, is there a higher concentration of a solute in which the membrane is impermeable is called osmosis. Figure 123. Diagrammic representation of osmosis. Water molecules are represented by small open circles and the solute molecules by large solute circles. In the diagram on the left, water is placed on one side of the membrane, permeable to water but not to solute, and an equal volume of solution of the solute is placed on the other. Water molecules remove down their concentration have to be applied to prevent the movement of the water molecules. In an important factor is physiological process. The tendency for movement of solvent molecules to a region of greater solute concentration can be prevented by applying pressure to the more concentrated solution. The pressure necessary to prevent solvent migration is the osmotic pressure of the solution. Osmotic pressure, like vapor pressure, lowering freezing point, depression, and boiling point elevation, depends on the number rather than the type of the particles in a solution. That is, it is a fundamental colligative property of solutions. In an ideal solution, osmotic pressure P is related to the temperature of the volume in the same way as the pressure of a gas, P equal to nRT by V.
Of plasma tonicity. The freezing point of normal human plasma averages minus 0.54 degrees centigrade, which corresponds to an osmolar concentration in plasma of 29 mole osmol per liter. This is equivalent to an osmotic pressure against pure water of 7.3 atmosphere. The osmolarity might be expected to be higher than this because the sum of all the cation cation and anion equivalents in plasma is over 30 millimosmol per liter. It is not this high because plasma is not an ideal solution and ionic interactions reduce the number of particles free of exit and osmotic effect. Except when there has been insufficient time after a sudden change in composition for equilibrium to occur. All fluid compartments of the body are nearly in osmotic equilibrium. The term tonicity is used to describe the osmolarity of solution relative to plasma. Solutions that have the same osmolarity as plasma are solid to be isotonic. Those with greater osmolarity are hypertonic, and those with lesser osmolarity are hypotonic. All solutions that are initially isometric with plasma that have the same actual osmotic pressure or freezing point depression as plasma would remain isotonic if were not for the fact that same solution diffuse into cells and others are metabolism. Thus, a 0.99% saline solution remains isotonic because there is no net movement of the osmolarity active particles in the solution into cell into the particles are not metabolized. On the other hand, a 5% glucose solution is osmotic when the initially infused intravenously.
Equivalence. The concept of electrical equivalent is important in physiology because many of the solids of the body are in form of charged particles. One equivalent EQ is 1 mole of an ionized substance divided by its valence. 1 mole of NaCl dissociates into 1 EQ of Na plus and 1 EQ of Cl minus. One equivalent of Na2 plus is 23 grams, but 1 EQ of Ca2 plus is 40 grams, 20 grams. The molecular equivalent is 1/1000 of EQ.